Hello, students. This is Dr. Faizan Mirza. In this video, what I'm doing is I'm trying to cover some P4 questions regarding gene technology and the chapter 8 for A2 biology. Now, this question that I've taken some three, four questions here that I want to discuss. Let's start with the first question. This question deals with this question is from this question is basically from October November 2014, and this is variant 43. And this question basically is about the question asked. The diagram outlines how a gene coding for human insulin is produced by genetic engineering techniques. Step one, two, three are provided to you. Step one shows to you that mRNA of the human insulin gene is being taken out, then something X is actually being added, and step two is actually following this, where a CDN is being made. We know CDN is a single stranded DNA. So X must be the enzyme reverse transcriptase, because the reverse transcriptase enzyme, it basically converts this your mRNA into a cDNA molecule. Then from step two, we move on to step three, where using this thing Y, which will be the DNA polymerase enzyme, you're converting the cDNA into a double-stranded DNA. In this case, the single-stranded DNA is made into double-stranded DNA. So this must be DNA polymerase. So gene is prepared. In step three, the gene is ready for you. Then step four, they are saying that additional non-coding DNA is added. This additional non-coding DNA could either be the sticky ends being added or the promoter being added. It could be either way. They have not specified, so we won't go there. But if they would have asked, so we could have answered that step four is basically about sticky ends or the promoter. Then they're asking name the enzymes X and Y. So we already have listed the enzymes, reverse transcriptase and DNA polymerase accordingly, so we can mention those. Then they're asking us about the explain the starting why the starting point of this procedure is an mRNA and not a DNA. So we know that mRNA is in the cytoplasm and it's in large quantities present in the cytoplasm, and it's for one gene only. You don't have to cut the mRNA because it's already of one gene having the whole amino acid sequence code on it. So DNA extraction, on the other hand, would have been very difficult because it's in the nucleus and only two copies of the gene would be present inside the nucleus. The next question actually takes you to the to suggest, where they're asking you to suggest why some methods of manufacturing genetically engineered insulin use eukaryotic cells rather than prokaryotic bacterial cells. So basically, you are asked to compare eukaryote against a prokaryote. So we know that eukaryotic cells, yeast is an example of eukaryotic cells. So it would have the rough ER and the Golgi for protein synthesis as well as the modification, respectively. So insulin, which is being produced using yeast, would actually be of appropriate three-dimensional shape, similar to the human insulin, because human cells are also eukaryotic cells. Whereas if the insulin was being produced from bacterial cell, it would not be modified the way we expect it to be modified in a eukaryotic cell. Stage three advantages of using human insulin produced by genetic engineering. So there are no allergic reactions because insulin is not taken from the cattle, no ethical and religious issues, cheap and not very expensive, large quantities can be produced. So we can add these points here. Acha, one more thing. At time I've seen the students being confused about how we go about with genetic engineering. So if you're using the human tissue, so we'll take blood for example, and or not even just human, for animals as well. We'll take blood, and from blood we take WBCs, because the WBCs are the cells that have the nucleus. From there we can extract DNA. The DNA that we extract for gene technology, this we are trying to extract the gene from the DNA, or we are trying to identify a gene. So what we'll do, we'll just extract the DNA from the viral cells, we'll fragment it using restriction enzymes, but cut the DNA into different fragments. We'll amplify these fragments using polymerase chain reaction or PCR, and we'll apply the current and the potential difference on the gel. So we'll follow this with gel electrophoresis for separation and identification of the gene, although not very related to what we have just discussed, but there's something, a mind map that I've just shown here, because I've seen students being a bit confused about how we start up with extracting, or what protocols do we have to follow when we are using this genetic technology experiments. Let's move on to the next question. Okay, so this question is from October, November 2016, variant 42, and the question basically is about PCR. We know that PCR is used to produce large quantities of DNA from small original sample. The main series of PCR are shown in figure 3.1. There are three steps shown, step one, two, and three. We know step one is about heating the DNA sample to 95 degrees centigrade. We call this as denaturing, where a DNA, which is a double-stranded structure, we know that double-stranded DNA, it breaks up. The complementary base pairing, the hydrogen bonds, they break, and we have 5' to 3', one strand separated from 3' to 5', the other one. So this is denaturing. Then step two, which is we cool it down to 65 degrees Celsius. We have these learned facts with us. Primers are added, and we call this step as annealing. So how the primers are added? Basically, this strand, which was 5' to 3', this was the one, and the other strand 3' to 5' is this, which is being separated. So these are two single strands we have. We add a primer to the 3' end of both. The primer then allows DNA to get elongated up along the appropriate direction, because we know that for the DNA to elongate, we need the enzyme DNA polymerase, and the DNA polymerase only works on the double-stranded ends. For example, if we wanted this DNA polymerase to start working on this single strand, it just would not work, because the DNA polymerase cannot work on single strands. It needs a double-stranded end to start working on. So that's why we add primers. And that's a process called annealing. Then we move on to step third, in which we heated this to 72 degrees Celsius. Taq polymerase is added, and DNA is synthesized. We call this process elongation. So this is one cycle of PCR. When you repeat these cycles over and over again, so DNA gets amplified. They're asking you about explain why DNA sample is heated to 95 degrees Celsius. So yes, we mentioned about the hydrogen bond being broken, about the complementary base pairing being separated, the DNA strand being separate from one another. So both the strands can be used as a template. Then they're asking why primers are added. Explain. So when we explain why primers are added, so we know both strands are single-stranded. So at 3' end, we attach the primer. So now that end becomes a double-stranded end. And to this double-stranded end, DNA polymerase can later attach, and DNA elongation can continue. DNA polymerase can only attach to double-stranded ends. That's the catch here. Explain why Taq polymerase is used in step three. So we know Taq polymerase is a heat stable DNA polymerase. It will not denature at high temperatures. And so we don't have to top it up again and again during amplification and heating cooling cycles. The next part, they take you to selection evolution, in which we study about mitochondrial DNA. And they say that once an organism dies, the DNA gradually breaks down. However, the cells in the bones that are buried hundreds of years ago may still yield small amounts of DNA that can be extracted, amplified using PCR, and then analyzed. Mitochondrial DNA is often used because there are usually more than 100 copies of it in one cell. Why? Why you have more than 100 copies of mitochondrial DNA? Because there are many mitochondria in one cell. So every cell may have as many as 100 mitochondria, so there can be 100 copies of mitochondrial DNA in one cell, compared to only two copies of nuclear DNA, because we know that inside the nucleus, the DNA is basically of a diploid cell. So one gene will have like two copies, two alleles there. For example, in 1994, mtDNA from the bones had been found in grave in Russia, was analyzed to confirm that these remains of the royal family, who were known to have been killed in 1918. The mtDNA extracted from the bones were compared with the mtDNA from a living relative. The family pedigree is shown. Now, look at this pedigree. We call this kind of a chart as pedigree. So there's this Queen Victoria, and this is the symbol that whenever we see a pedigree, we basically just first of all look at the symbols. A circle represents female, and a square represents male. So wherever you have circles drawn, these are all females, and wherever a square is drawn, that's a male. So we know that mitochondrial DNA passes along the maternal lineage. So if this person, the living relative male being this person called G, this person must have received the mtDNA from the female F. The female F must have received it from the female above her, like in the family tree. And both these females must be siblings. That's why they basically have a common mother, and this is the common mother, and they have the same grandmother as well. So this lady, Queen Victoria A, passed on the mtDNA to this lady, and she passed it on to both her daughters. This daughter then passed it on to her daughter F, and it eventually came to this individual G. Now this person C here gets married to B, and they have these many kids. How many of these will have the same mtDNA as Queen Victoria? All of them. Why? Because they all will receive the same mtDNA from the female lineage. So in this particular chart, the person Nicholas B is the only one who will have mtDNA from a separate female line, whereas all others have the same mtDNA because we all actually are the descendants of the same woman. Explain why there are usually more than 100 copies of mtDNA. So we have already covered this. We can just summarize this here. All mitochondrial DNA come from the egg and not sperm, maternal lineage. So we just need to mention which letters have the same mtDNA to relative G. I think that's very simple. And with this, we can move on to the next question. Okay, so this question here is from May June 2019, variant 42. Soybean, glycine max, is an important food crop for human consumption and for feeding of animals. Two varieties of soybean, Winton 81 and GTS 4032. This Winton 81 has been developed in a traditional way by selective breeding, which means artificial selection, whereas the other one is by genetic modification. So there are two varieties. This one, which is genetically modified, is herbicide resistant. It can withstand application of glyphosate herbicide. Glyphosate is sprayed on the crop to kill weeds. Outline has genetic engineering that gave this particular variant the trait of herbicide resistance. So what would you do? We will take the resistant allele. The gene for herbicide resistance taken from a species other than soybean. If you can recall the name Agrobacterium very well, if you cannot, still you will get the mark here by mentioning that the gene is taken from the other species, other than the soybean. Restriction enzyme is used to cut the plasmid, and the resistant gene is added to the plasmid. The promoter is then inserted together with the gene. Because we know the gene will not switch on until the promoter is there, we add this GFP gene so that screening is possible. And as a marker, the transformed cells are then screened and grown in culture. The recombinant DNA added to the soybean. Now, when this is added to the soybean, the gene gets delivered and it gets expressed inside these cells. The product of this gene stays unaffected in the presence of glyphosate. So you basically summarize all the procedures that actually went on to make this organism herbicide resistant. So, and you have to outline, you don't have to give a detailed description of. Countries vary in extent to which they grow GM varieties, such as this. Traditional varieties, Winton 81. They have given two conditions here. In USA, 94% of soybeans are genetically modified, so you have more of genetic modified organisms grown in US. In Europe, 0% of genetic modified are grown, so basically not grown there. Scientists use computer models to predict the effect of two different changes in agriculture practice on soybean crops. So these are computer-generated models. A global ban that reduces cultivation of soybean to 0%, which means something similar to Europe, if that's done globally, and all countries increasing cultivation of GM soybean like US. So these both conditions are being replicated in a computer-based model, and prediction is being actually it's being predicted using the computer model that how it affect. So table 3.1 show the results of this modeling for 4 countries. So if you apply both these on these 4 countries, Europe model and the US model. So what do we see? We see the percentage change in the yield of soybean. So we see that the yield is not affected in these 2 countries, but it reduces in the other like Bolivia and US. So it overall shows a decrease. But if US model is used, so we see no change in US and Bolivia, but we see a positive change or increase in the other 2 countries. Explain what the data in table 3.1 suggests about the social and ethical implications of growing GM soybean. Social and ethical implications are what is being asked. So if we ban GM soybean cultivation, like the Europe model, it would lead to overall decrease in the yield because that's what's shown. The overall yield will decrease. Whereas increasing the cultivation would increase the yield. With higher yield, more food supply is available. So if you follow this, more food supply will be available. This can help reduce starvation because we have to basically address social and ethical applications here. In the poor countries, although would cause a higher use of herbicides, yes, because we are using more of herbicide now. Since genetically modified organism is being used, you will need more herbicide to kill the weeds. So more money will be spent on the herbicide. Long-term consequences of health cannot be predicted. Frequent use of soybean, this genetically modified, can reduce the biodiversity and can affect the biodiversity in some negative way. Moving on to another question. This question came in Feb March 2023, waiting for you too. And this question basically deals with microarray. So, edit comparative genome hybridization. They have given a technique ACGH, a new technique that we do not study, but we do know what hybridization means. Hybridization is simply two strands of nucleic acids binding to one another, complementary sequences. So it's a technique involved using microarray to analyze the genome and or the sections of the genome. Either the whole genome or just the part of that genome is being basically monitored here. Outline the steps required to prepare genome of individual so that the genome is ready to analyze the microchip. So what do? You extract the DNA. And how do you extract the DNA? So you extract it from the white blood cells, the blood, and when you take those out, you fragment it using the restriction enzymes. You denature it. You denature it means you separate the two strands by heating, and the two strands get separated because for hybridization on microarray, we need a single stranded DNA fragment. We cannot add double stranded DNA. So you need to denature the DNA and heat it to separate the two strands. Then add fluorescent tag and add it to the microarray chip. So that's what we do for adding the genome sections to the microarray. Now this question moves to DiGeorge syndrome. It's a dominant inherited disease in humans. DiGeorge syndrome is caused by deletion of large number of nucleotides from chromosome number 22. So large nucleotides get deleted. The number of deleted nucleotide varies between individuals from 800,000 to 3,100,000. So that's a huge range. The largest deletions can cause the removal of up to 46 protein coding genes. So separate 46 protein coding genes can be removed from the chromosome 22 altogether. Figure 4.1 show the results of ACGH using microarrays specific for a section of chromosome 22 with the DiGeorge syndrome. Deletion occurs. Microarray analyze DNA of two individuals. One is having DiGeorge syndrome, the other is not having DiGeorge syndrome. The person who's not having DiGeorge syndrome is the control DNA for comparison. In the ACGH results shown in figure 4.1, these four bullets are what we need to understand. Each small circle represents the result from a single probe on the microarray. We know the microarray gives many probes, so at every point you will have hybridization occurring wherever the gene is present. The x-axis shows the position of each probe on the chromosome 22, the relative position of the loci. The position is shown the distance along the chromosome in millions of nucleotides. A result close to 100% fluorescence on y-axis means the DNA from the individual with DiGeorge syndrome fluoresces at the same intensity as the control DNA. So both DNA fluoresce 100%. So that's the control DNA as well as the DiGeorge syndrome. So they both have the same sequences there. That's why 100% is being fluoresced. A result of close to 50% fluorescence on the y-axis means the DNA from the individual DiGeorge syndrome fluoresces half as much as the control DNA, which means that this 50% fluorescence shows DiGeorge syndrome. It's not fluorescing exactly at the same level as the control DNA because the control DNA will not have any deletions. This is the table, or you can say the graph shown. These are the relative positions mentioned. Millions of nucleotide on chromosome number 22. And this is 100%, this is 50%. So every circle represents the probe here and its relative position. So all the 100% ones, these are the ones which are common in both. The 100% ones are the ones which are control and DiGeorge syndrome. Both individuals show this. Whereas these fluorescence show the ones which are which actually have been deleted in the individual who is suffering from DiGeorge syndrome. These have been deleted. That's why the fluorescence is not the same as the control DNA. The question that they're asking, although just for 1 mark, is with reference to figure 4.1, estimate the number of nucleotides deleted from the affected chromosome 22 in the individual from DiGeorge syndrome. Give your answer to the nearest 100,000 nucleotides. So what we'll do, we'll just count the number of squares. So if you look at this graph here, so this is where it begins and this is where it ends. So what we'll do, we'll just count the whole squares. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, and including these two will make it 25. So our answer will be to the nearest 100,000 nucleotide will be 2500,000. Explain how the microarray technique works to give the results in figure 4.1. How it works, the probes of microarray would have the sequences that can identify the allele of chromosome 22. The DNA is collected from the DiGeorge syndrome and the normal control cell. Both are labeled with different polar fluorescent tags. Allow them to hybridize in the microarray. Wash the unhybridized fragments. This is the mandatory part because we have to wash off the unhybridized fragment so that everything will not fluoresce. Compare the fluorescence of individual DiGeorge with control. Control DNA binds at 100% probe, whereas the DiGeorge binds to 50% probes. Last, suggest why the phenotype of two individual DiGeorge syndrome can be very different because they already have told us that the deletions can be small, like can be in the range of this thing from 800,000 to 3,100,000. That's a pretty long range. So they're asking that how could these two individuals be different? Different individuals have different organs, different set of deletions of base pairs. We can expect that both would have different number of genes being deleted because up to 46 genes can be deleted. This can be an example of continuous variation. So both individuals might be belonging to different environmental conditions because we know that in continuous variation, environmental effect is seen on variation. So these were the question that I wanted to discuss. I hope this video was helpful for you. That's it from my side. Thank you so much.